graduate student in the Department of Genome Sciences here at the NOD Lab, and I'm going to talk today about how conflict and coevolution shape the evolution of the primate kinetophore. So centromeres are conserved and essential at every cell division. For instance, in mitosis, the centromere is the primary constriction point of the chromosome. Kinetophore proteins recognize and bind the centromere, and then microtubules attach there, ensuring that the sister chromatids are properly segregated to two daughter cells, and each daughter receives a full complement of genomic DNA. So the centromere is essential in every cell division, and its function is conserved throughout all eukaryotic evolution. But centromeric DNA evolves quite rapidly, which is not what we'd expect from a conserved element, or a functionally conserved element. For instance, in budding yeast, they have a small, relatively simple point centromere that's specified by the sequence of the nucleotides. But that's not common necessarily even within fungi. For instance, the other model fungi fission yeast has a much larger, more repetitive centromeric sequence. In addition, in plants and animals, centromeres are primarily made up of repetitive satellite DNA. But even there, the structure and sequence can change quite rapidly. For instance, gibbons and humans are both hominoid primates, but gibbon has a very different arrangement of its alpha satellites that is a monomeric repeat in which each satellite is highly identical to its neighbors. Whereas human, although still centromeres still are comprised of alpha satellites, they're arranged in alpha satellite arrays in which the satellites are not necessarily identical to their neighbors, but highly identical to satellite in the same position of the array, potentially many repeats away. In addition, plants can have show an example of an even more complex centromere, where they have repetitive satellites, but also centromeric repeats and even transcribed genes in the centromere. So, why are centromeres rapidly evolving despite the conserved function in cell division? And the Math Lab has a model for why this is happening. We call it the centromere drive model. So when we see rapid evolution despite conserved function, we immediately think of genetic conflict in the Math Lab. Genetic conflict is able to produce repeated episodes of positive selection despite conserved function. And one of the canonical examples of genetic conflict is that of the host pathogen conflict. So what's important here is both the host and pathogen genomes have opposite goals. In order for the pathogen to survive and replicate, it must infect the host and at least lower its fitness, if not outright destroy the host. So the host encodes defense proteins that recognize and hopefully restrict the pathogen. When this host protein is able to recognize the pathogen and restrict it, the host is winning this conflict, and the pathogen must find a variant in the population, or a mutation must arise that can avoid recognition by this host protein and escape the restriction and allow the pathogen to infect the host. This puts pressure back on the host to evolve to recognize this new pathogen protein. What's important here is that this puts pressure back on the pathogen in order to evolve again and back on the host. And there's no global optimum. There's no way that evolution can find an optimum for this host protein for all pathogens. What the best this pathogen can do at any given point is based on what the pathogen is doing at the time. So this is why you get repeated rounds of evolution despite this basically conserved function. And this is called the Red Queen hypothesis. From the Through the Looking Glass by Lewis Carroll, the Red Queen says it takes all the running you can do just to keep in the same place. It takes all the evolution that this host can muster just to maintain that interaction. So these host pathogen conflicts can be detected through signatures of rapid evolution. This region of the protein is changing quite rapidly in order to recognize the pathogen. So rapid evolution can not only detect this, but it can provide insight into the interaction interface between the host and the pathogen. The evolutionary theory is that only the interaction interface will be rapidly evolving in order to maintain this recognition. So that begs the question, what conflict could the centromere be involved in that could be driving this rapid evolution? I've already told you about mitosis and the function of mitosis to create two daughter cells genetically identical to the mother cell. And there's really no opportunity here for these two chromosomes to compete for any kind of evolutionary advantage in mitosis. So that's obviously not where the conflict involving the centromeres arises. So in male meiosis, similarly to mitosis, the centromeric DNA is replicated, or excuse me, the chromosomes are replicated, but in this case you go through 2 rounds of cell division in order to produce 4 haploid daughters. In male meiosis, it's symmetric meiosis, so all 4 of those daughters go on to become gametes. And these gametes are able to compete with each other. The green and the blue chromosome can compete for evolutionary success in this case. But importantly, this competition occurs after meiosis is complete. So the centromere, which is functional in the cell division, has much less opportunity, in fact, very little opportunity to compete in male meiosis, because all of these go on to become gametes. So female meiosis is a unique opportunity because it's an asymmetric meiosis. Just like in male meiosis, DNA is replicated and 2 rounds of cell division occur. But unlike male meiosis, the 4 haploid daughters do not all go on to become gametes. Only 1 of the 4 goes on to become the egg. The other 3 are discarded. So in this cell division, the blue chromosome was preferentially positioned at meiosis 1 and made it into the egg. But if the green chromosome is able to flip the tables and position itself properly at the meiosis 1, then the green chromosome can win. So that creates a competition between this blue and green chromosome for positioning at meiosis 1 in order to reach this egg.